The ERMAC Centre is proud to present the SFU Canada Research Chairs Seminar Series. This bi-weekly series hosts 6 presentations per semester. For the fall 2008 and spring 2009 semesters, the presenters belong to the Faculty of Science and the current Faculty of Applied Science. Today's speaker is Dr. Randall M. Peterman. Dr. Peterman will present his talk entitled Research for Improving the Understanding, Conservation, and Management of Pacific Salmon Populations.
It gives me great pleasure today to introduce Randall Peterman, who will be talking on research for improving the understanding, conservation, and management of Pacific salmon populations. Randall was actually a founding member of the School of Resource and Environmental Management. He's been here for almost 30 years, which is rather remarkable, and has made enormous contributions to SFU and to REM during that time. But today we're here mainly to focus on his external contributions through his research. We often in REM talk about the importance of using our research to address critical problems in the management of resources and the environment. But the flip side of that we also try and think about is trying to look beyond today's critical problems to identify and help define what will be the critical problems of the future. And in trying to think about what was it that I wanted to say in introducing Randall, one of the things that really strikes me is how successful he's been at not just that first task, but that second task, in his ability over the past 30 years and more to really anticipate where we needed to be going and define the agenda for what we're going to be trying to do in fisheries management. When I think about issues that have become central, that he played a key role in either defining de novo or bringing to the fore in fisheries management and resource management more generally, there's a pretty impressive list. Things like bringing statistical power kind of from the bowels of statistics departments into kind of practical usage for understanding how we interact with resources, the precautionary principle, bringing Bayesian methods kind of into a standard usage within ecology, bringing careful and critical thinking about risk and uncertainty into its central place in resource management today. And kind of probably the biggest of all, kind of the role of adaptive management in how we think about how society interacts with resources. And Randall's had an absolutely critical role in all of these areas, which has been inspiring to all the rest of us in REM. His impact in this regard is reflected in many ways, but just an easy example is in citations. In 2007, 2008, his published papers had almost 400 citations. And when you bring in a book that he was a co-author in that was published in 1978, a long time ago now, that bumps up to over 500 citations in 2007 and 2008, which is remarkable. But he's not just a fantastic and influential researcher, he's also an excellent instructor. He's won SFU's teaching award. He's been an extraordinary mentor and team builder, especially within the fisheries group at REM. He's built it from just himself in 1979 to a large, strong, and internationally recognized research team. And he was the founder and director of the Cooperative Resource Management Institute. Randall did his PhD at the University of British Columbia and has been here most of the time since then. He actually got his start working on forest insects and made significant contributions there before switching to fisheries. And I hope you'll join me in hearing what he has to say and welcoming him to this presentation. Thank you.
Well, thank you very much, Ken, for that very nice introduction. I'm very humbled. And if I were you in the audience, I'd be thinking, this better be good. Okay, so most people start out talks, most people end talks with acknowledgments. I'm going to start one with the acknowledgments because I want to put up right up front that the main contributors to the research I'm going to talk about today are my former students, postdocs, research assistants, and current ones, and they should get as much credit for this work as I do, if not more. I've also managed to benefit from the wisdom and experience of other researchers, other faculty members, researchers at management agencies, and visiting faculty as well. I've gotten grants from a number of the usual sources and have cooperated with people in various management agencies, and they have kindly provided data of the sort you'll see today. So at the core of what I do in my group is salmon ecology research, but we apply it to conservation and management. So I'm going to talk today about several examples that draw in methods from all these different fields: oceanography, climatology, statistics, medicine, engineering, and business. And I'll give examples of each of these in order. So starting with the core of salmon ecology, though. You've all lived long enough here on the coast probably to realize that Pacific salmon are important in many ways to commercial and recreational fisheries, to First Nations, and to tourism. It's actually an icon for the quality of the environment we live in. Now, just to refresh your memories, in case you haven't absorbed this through newspapers and such, adult fish spawn in fresh water. Their eggs hatch into juveniles, about 1 to 10% of them on average. Those juveniles then migrate into the ocean, where depending on the species, they will migrate out into the Gulf of Alaska, mingling with populations from other regions. The fish that are in the ocean will survive, say at a rate of about 1 to 10% on average, to come back as what are known as adult recruits. And so these are the fish that are produced and survive from the eggs. Some of them are harvested, some make it back to spawn. And what you're going to hear a lot about today is this ratio right here of adult recruit abundance per spawner. Recruits per spawner is what I'm going to call productivity. And so this is looking at how productive are individual populations. We're asking, are they very productive, or are they just barely hanging on, or are they decreasing? So just to take a quick look at the status of Pacific salmon stocks around the Pacific Rim, at least on this side of the Pacific Rim. Here are wild populations, not hatchery fish, not aquaculture fish, in thousands, for 3 species for which we have long data series. You can see that there are some populations like this one in northwestern Alaska that's going down precipitously over time. Another of the same species is going up. Pink salmon in southern BC, quite variable, large abundance, and then others that are just staying down at low abundance. Sockeye going up here in the Wood River system, dropping fairly dramatically over the last decade or so in southern BC. So there's quite a variable description of the status of these populations depending on where you are and what species you're talking about. And these are just, of course, some examples of the many hundreds of populations out there. Now, one other thing to note is that these populations are highly variable from year to year. Not just the long-term trends are variable across stocks. So this variability has implications for the conservation, management of harvest, and for the users of these fish. So the 2 broad research goals that we've had in my research group for a long time are to, first of all, increase the understanding of fish population dynamics in general, and to develop and test various methods to improve that understanding, as well as to provide information to resource managers and conservation groups. So the ultimate aim, of course, is to improve the status of salmon populations. So I'm going to talk first now about some examples where we can apply some knowledge of salmon ecology using information on oceanography and climatology to conservation and management. Our research objective in this first example was to understand the causes or sources of variation in productivity across time. And productivity, again, is measured as adult recruits per spawner. So keep that in mind. It's how many offspring survive to become adults per spawner. Now, why is this productivity important? It's because it affects the response of populations to harvesting. The more productive a population is, the larger portion of the adults you can take and still maintain a constant population. And more likely it is that a population could absorb the detrimental survival conditions that nature throws at them. The rapidity of recovery from a depletion is more rapid with a more productive population, obviously. So we need to know productivity for these 2 reasons. So the typical method for understanding the environmental causes of variation in productivity are to do what we call shotgun statistics, and that is you take the data on the biology and every possible environmental variable you could get, you throw them into correlation matrices and see what pops out, and you write a paper on the significant cases. And of course, what that leads to is many spurious relationships that as time goes on, the data are no longer consistent with that initial prediction, so that fails to work. We took an alternative approach, which was to ask the question, is there variation in productivity that is shared among separate populations? Because that shared response will provide a much stronger signal of some effect than you get if you looked individually at populations. So in essence, we're asking, is there a positive correlation in productivity among salmon populations? And in other words, is there a spatial synchrony? So over the decade of about the 1990s, we put together a salmon database for 120 stocks throughout the western North Pacific, and they averaged about 31 years in duration. And their data on spawners, which I'll symbolize for this talk as capital S, and adult recruits, R. And so what you see here for 3 different species is the location of the point of ocean entry for the juveniles when they migrate into the sea. So there are 43 different populations for pink salmon, 37 for sockeye, and 40 for chum salmon. What we did for each one of those populations first was to remove the within stock density dependent effect. And the reason we needed to do that is if we just looked at log recruits per spawner over time, you can see that as spawner abundance changed, you would get variation in log recruits per spawner that had nothing to do with environmental effects or effects of predators or food supply. So we want to remove that within stock effect first, and we calculate, so each one of those dots is a different year of spawning, and we wanted to calculate the residuals from that within population effect for each of the years. And so we generated a series like this of residuals for each population. And these residuals are in units of log recruits per spawner. So you can see in this graph that in the early 1970s, the population was much less productive than normal for whatever reason. And so we did this calculation for all 120 populations, and then we asked how well correlated are these residual time series. So this is for pink salmon in northern Alaska, 2 different populations. They're located here in the Alaska Peninsula, and these are the 2 time series for those cases. You can see a high positive correlation among those 2, and these are standardized, by the way, just to have mean of 0 and standard deviation 1, just for graphical purposes. So we did the pairwise correlation among all possible pairs of the 43 pink salmon stocks, and we plotted those correlations as a function of the distance separating their points of ocean entry. And that's this graph here. So this point right here would be a correlation between 2 stocks that were separated by about 500 kilometers in terms of their points of ocean entry. And what you see here is that there's a positive correlation when the populations have close points of ocean entry, and this correlation tends to drop towards 0 when they're separated by a distance of about 1,000 kilometers. Now this correlation function can be described more simply in terms of a simple single metric. And what we did here was to say, at what distance does that correlation drop to half of its maximum value? So the maximum was 0.55. It dropped to half of that at a distance of 470 kilometers for the pink salmon. And in a map, what those regions of positive correlation look like are these blue ellipses. So when conditions are good for one population, they're good for other populations within that blue ellipse, but not necessarily good for the populations in other ellipses. So we repeated the same type of spatial correlation analysis for the chum and sockeye salmon. I just showed you the results for the pink salmon. And all 3 of them came out with a correlation distance, a half maximum correlation distance of between 500 and 800 kilometers. Sockeye was about a little over 500. Chum was just a little bit less than 800. So all 3 species are showing a similar spatial scale of covariation. And this is completely contrary to what the previous research had suggested, which was that there's a North Pacific-wide positive correlation in the order of the scale of thousands of kilometers. Unfortunately, those analyses were done with catch data rather than biological productivity. This is a much more appropriate index to use. So the question is, where does most of this covariation among populations in their productivity arise? Well, I don't have time to go into the evidence, but there's very strong set of evidence that it's late freshwater or early ocean life where this arises. So what's driving this spatial covariation in productivities? Could it be just that we're counting dispersal or straying among populations? Well, no. We can identify individual populations very easily from their scale patterns, their parasites that they carry, and this sort of thing. So maybe it's predators. Maybe they're predators that are causing significant changes in productivity up or down, depending on where they're spread and the populations that run into them. That's possibly true, but unfortunately we have almost no data of a long-term nature for very many locations for the predators. Same thing with the food. It could be that the food supply is short or abundant over large areas at the same time, thereby affecting many populations at the same time. That's quite possible, but again, we don't have data for very many locations, let alone for much time. So this leaves us with using environmental factors as either a direct index of change in productivity or an indirect indicator of predators and food. And it's the latter that you'll see shows up here in a bit. Okay, so now rather than doing the shotgun statistics approach of asking which environmental variables are causing this variation in productivity, we applied a screen to find the candidate environmental variables that would be most appropriate to use in analyses. So we asked which environmental variables have a similar spatial scale to that of the productivities that we're trying to explain. In other words, we're going to look for environmental variables that have that spatial correlation index of about 500 to 800 kilometers. So we have long time series at many different spatial locations for 3 environmental variables, upwelling, coastal sea surface temperature, which I'll designate as SST for the rest of this talk, coastal sea surface salinity. So we did exactly the same type of spatial correlation analyses as we did for the salmon with these time series of environmental data, looking at the distance between the monitoring stations. What we found was that the early summer sea surface temperature was the only environmental variable that had the same spatial correlation scale as that of the fish productivity that we're trying to explain. So that suggested to us that this is a candidate variable we should have a more serious look at and try to develop models that include it. So that takes us to the next step, which is using some statistical methods to examine the extent to which SST and other variables perhaps influence salmon dynamics. So our research objective was specifically, does sea surface temperature help explain variation in salmon productivity? And the method that we used was to simply add SST that was specific to each stock's location to various models, and using SST as an environmental explanatory variable. So these are stock recruitment models. That's the shorthand notation for the relationship between the number of spawners and the number of adult recruits produced. And by the way, I use the word population and stock synonymously, as we do in this field. So one of the big challenges to doing this type of analysis, though, is that we've got large observation error in the abundance of spawners. We can't ever count them exactly. So that observation error means that we have imprecision in our estimates. And if you know anything about drawing up statistical models and estimating them, you need to know what the form of the error structure is and take that into account in your method. Furthermore, there's unclear model structure in terms of the biological function. So quite aside from the noise. So we actually tried out 24 different types of models with different assumptions. And here's what we found. For the 8 models that did not have any sea surface temperature variable included in them as an explanatory variable, we had this amount of unexplained variation. For those of you in statistics who don't recognize this, DIC is a measure used with Bayesian hierarchical models. And a lower value indicates better fit. So in contrast to this, every model, all 16 of them that included sea surface temperature, had better fit to the data than the models that did not include SST. And the 2 best models were what we call multi-stock spatially structured models, which I'll explain now. Conceptually, what a multi-population model does is it takes account of information from several populations at once. So what we're trying to estimate here now in these models is this parameter I'll call gamma. It's a I is the stock from 1 to 43 for pink salmon. It's the change in salmon productivity per degree increase in sea surface temperature. And so what you can think of in a multi-stock hierarchical model is that that parameter is drawn from some distribution that's shared among the populations. There's some unknown mean and variance in that distribution. And so we estimate these gametes for each of these populations. Now, the advantage of this approach is that it uses the fact that we've got positive covariation among the populations. So they're essentially statistical replicates in this kind of model. And furthermore, the observation errors that arise individually in population tend to get averaged out when you put them together. It doesn't mean they're zero, but they're reduced from what they would be if you treated each population separately. So the result of applying this analysis is the following. Here's a graph that shows the estimate of the effect of sea surface temperature on productivity on the x-axis. So this is the change in salmon productivity in units of log recruits per spawner per degree centigrade increase in that sea surface temperature in the summer. And then along this axis are the 43 pink salmon stocks arranged from south to north. Now first let's just look at the open circles. The open circles represent the estimates of this parameter that would have been obtained if you just took the data set for each population separately and you ignored all the others. You do analysis with population 3, you ignore all the others, you finish it, do the analysis with population 4, and so on. The multi-stock analysis estimates the parameters for all 43 populations simultaneously. It's a mixed effects model. And what you get is a much better estimate of the average effect of each degree change in sea surface temperature on the productivity. So here you see this line right here separates British Columbia from Alaska, by the way. So those fish stocks in Alaska have a strong positive effect of increasing sea surface temperature, whereas there's a negative effect in British Columbia. This general pattern was shown for the other two species as well. In the north, there's a positive effect of increasing sea surface temperature on productivity of chum and sockeye salmon, a negative effect in the south for sockeye, but nothing different from zero for chum. So what this analysis shows is that compared to single stock analyses, multi-stock models actually provide an improved understanding of the environmental effects on salmon productivity. Furthermore, these improved estimates of productivity, or the Ricker A parameter for those familiar with that, knowing that better will help reduce the chances of overfishing the populations and to increase the chances of recovery from depletion, because we'll know how much we need to relax fishing in order to get them to recover. Now, you're probably wondering about this bottom result here. Why is this negative and what implications does it have for BC? So this again says that for every degree increase in sea surface temperature in the summer, you get a negative 0.2 value log recruits. That turns out to be about an 18% decrease in the average adult recruitment. We think, we're not sure, but we think it's because of the following. When we have this west wind drift current coming into the coast, it bifurcates into a northward flowing Alaska current and a southward flowing current that becomes a California current. When that location of the bifurcation point moves north, and it moves north and south several hundred kilometers from one year to the next, when it's north, we tend to get warmer water from the south, which is poor in zooplankton, poor in food for salmon, and rich in predators like mackerel. So the southern stocks, British Columbia stocks, get hit with high predation rates, low food supply. So that's why an increase in sea surface temperature is associated with a decrease in productivity for those stocks. The increase in temperatures, by the way, are not enough to reach the lethal range. So it has nothing to do with the physiology. It's everything to do with an indirect index of what's going on in the ocean as indexed by temperature. In the north, when the average sea surface temperature goes up, that's associated with this northern movement of the bifurcation point. And what that does is the advection of food supply that's generated in the Alaska gyre through the spinning up of the central gyre gets advected onto the coast, where these fish are migrating along the continental shelf. So they have a greater food supply in years when there's warmer sea surface temperatures. So what does this mean for BC? The trend has indeed been for an increase in sea surface temperature over time here. And if you look at what implications that has for the survival rate of fish, the productivity has indeed gone down. That's a set of dots that basically led to the parameter estimates I just showed you. Now, you could easily take the step of saying, well, if temperature continues to increase, then obviously the productivity is going to continue to go down, and that's probably true. However, it's going to be hard to quantify that with any degree of confidence for the following reason. Sea surface temperature is actually only an indirect indicator of the biological system. Okay? It's not the lethal temperature. So these forecasts about the future are highly conditional upon the assumption that SST will tend to reflect the same sorts of biological conditions in the ocean that it has in the past. And that is difficult to justify because SST gathered right up here is a function of the interaction among solar radiation, wind-driven currents, and upwelling of cold, nutrient-rich water to the surface, which starts off the food chain that eventually gets to the fish. So there are many uncertainties in here about what SST is going to represent in the future, given climate change. Okay, so now let's go on to the next phase of the work, which is to talk about how we used a technique called decision analysis drawn from business to apply knowledge about salmon ecology to management of salmon. So this is some work that was led by one of my master's students, Lindsay Pestis. There's a conservation concern on this population. Cultus Lake sockeye is just up the valley here. We want to rebuild this population. You can see from the historical time series it's decreasing. It's down to a few hundred fish now, and it's in desperate straits. So the problem is that decline has been caused largely by fishing, but also by unfavorable environmental conditions. And these Cultus Lake fish are a small portion of a much larger group of fish that moves through the fishing area at the same time. These co-migrating stocks are ones you're familiar with, like the Adams River sockeye. They're extremely abundant and extremely productive, so they can be harvested at a high rate sustainably over the long term. But the cultus cannot. So if you're going to cut back on harvest rates in order to get the cultus stock to recover, that's going to have a dramatic effect on decreasing harvest rates on the other fish. So the research objectives for this project were twofold. First, define some harvest strategies that meet the recovery objectives for the cultus sockeye. And then second, to quantify the trade-offs between the salmon recovery, the cultus sockeye recovery, and the economic value of the harvests. Because this is what decision makers need, is a better estimate of the trade-offs. So the method we used was Bayesian decision analysis, which comes from business. Howard Rafer founded it in Harvard Business School about 40 years ago. It's one of many structured decision-making methods that essentially does a risk assessment for you by taking into account the uncertainties explicitly that are inherent in the system. It evaluates management options and provides a rank order of those options under a variety of assumptions that are usually evaluated through sensitivity analyses. Okay, in the model, we simulated two management objectives. First was to rebuild the Cultus Lake sockeye. And just as an example, here's a statement of a management objective. It's plausible. We would want a high probability that the populations would get up to 20,000 fish or above within 20 years. That's an ambitious goal, but that's one thing you could state as a target to model. And then we could ask, how can we maximize economic revenue of the commercial fisheries on the other co-migrating populations, not the cultus, as long as the first objective is met? So, to evaluate various harvest options, we did it in this way. This is a set of what are called harvest rules. This is the target harvest rate, the proportion of fish that are harvested on all the sockeye populations, cultus plus all those other much more abundant ones that they migrate with, as a function of the abundance of only the cultus stock. So the general shape here is intuitive. That is, you want the harvest rates to go down as the abundance goes down. And it's not clear just how much change you need to have in order to allow the populations of cultus to actually recover. This is a very heavy harvesting policy. This is a lighter harvesting policy compared to that one. And you can imagine lots of different shapes of these functions. And so in the model, you actually simulate the dynamics by having an estimate of the abundance at one time, and then you set the target harvest rate according to that rule. So we looked at 193 different shapes of harvest rules, just to cover the map of all possibilities. And I'm going to represent the components of this decision analysis in the standard format of a decision tree. So this square node here at the left is a decision node. Emanating from that are the alternative harvest actions. The first major biological uncertainty that we have is that you look at the data relating the spawner abundance to the number of juveniles, and it's all over the place. Huge scatter. So it's not clear at all what the underlying function is. Yeah, it's an increasing function, but what is the shape? It's likely nonlinear. So we fit a 3-parameter nonlinear function. And we examined a large number of possibilities by basically just saying, let's look at a range of those 3 parameters together and calculate with Bayesian analysis the joint posterior probability or degree of belief in each set of those 3 parameters. So this is one set, theta 1. This is another set that has a different shape of curve associated with it, and so on. So that is put in as the first uncertainty node, which is designated by a round circle here. As we've got all these possible shapes of that underlying function included in this analysis. So that's our first major uncertainty. The 3 ellipses, or the dots here, the ellipses, simply say that this uncertainty node with all of its branches apply equally to each of these other management options. We then weight each of those possible functions based on our degree of belief in them, which arises from the posterior probabilities that we calculated. The next major uncertainty is in marine survival rates. We know historically they're quite variable. We saw some of the data. And that's in log recruits per spawner. So poor, moderate, good survival rates. Again, those apply to all of these branches here. And then the third uncertainty, which most people have omitted up until a few years ago, is what we call outcome uncertainty. It's the extent to which the actual harvest rate realized at the end of the season deviates from the target. As you can imagine, there are many reasons why that deviation occurs. Non-compliance with regulations by the fishermen, biological and physical variation in the system so that the fish are down deeper than normal so they aren't as vulnerable to the gear, that sort of thing. So we represented this with another random process. And you can imagine that this decision tree would branch out into an enormous looking beast if we had put all those nodes together. But in fact, it's very quick to calculate all this in the computer. And having calculated the outcomes associated with each potential or each management action, following through each potential combination of uncertain states of nature with each action, we can calculate the indicators of recovery, namely the probability that the spawner is going to be above our target within the allotted time, and the average annual economic value of the harvest. So it turns out there are several rules that meet the recovery objectives. They're shown here. And the one that maximizes the gross economic revenue is the one in blue. So this says that it's only after the population gets above this amount that you could even turn on fishing onto the cultus and other populations. And after that, it should go up linearly to this peak. So now, if this is the key result of this work. A trade-off, a quantification of the trade-off that would be required by users and managers. So if the managers said they wanted a really high probability of getting recovery of the salmon population, namely 0.9, that means the average economic value from the commercial fisheries for all the fish stocks that co-migrate with the cultus would be less than $44 million. Or if they wanted to relax that and say, well, we only want 60% chance of getting recovery, then you'd have a little over $50 million. This result is an important example of what kind of quantitative information managers need to make decisions. We can't advise them on that. We just can tell them, here are the implications of your different decisions. Unfortunately, Lindsay's work finished 6 months after the minister decided not to list the cultus sockeye under the Species at Risk Act. So who knows whether it would have made any difference? It's hard to say. Okay, another element of risk assessment that I want, or another example of risk assessment that I want to show you about is some work that we did recently on populations in the AYK region. This is Western Alaska and the Yukon. So the Yukon River actually stretches from here into Canadian territory. The fish populations there have had quite a varied history, but mostly down. As you can see from these plots, these are 2 chum salmon populations, and in the late 1990s and early 2000s, they dropped so precipitously that the state of Alaska and the U.S. government declared that as an emergency disaster area, and they pumped a large amount of funding in to keep the subsistence fisheries going and the communities going. So we undertook some research with a grant from them to estimate the trade-offs among various objectives that included conservation objectives, commercial fishing, and subsistence fishing objectives. The model consisted of similar components to what you've seen and heard about in the past probably. We put together population dynamics models that included what's known as an Ali effect or depensation. This is where, as the population gets very rare, it becomes harder to get successful growth in the population, then it drops off towards zero very quickly, usually due to compensatory predation. There's also random variation in survival rates at various life stages. Fishing that was based on these harvest rules, like the ones I showed you for the cultist case. And then there was outcome uncertainty as well, to represent the variation in actual harvest rates compared to the targets. So one of my colleagues, Jeremy Colley from the University of Rhode Island, was here on sabbatical last year, and he worked with this along with Brett Zelke, who's in the audience, on co-op work term, to put together this analysis. And here's one of the ways in which results are represented. What you see here is a grid of cases that we simulated to represent different settings on the management dials, if you will. So we've got along the bottom axis here target spawners. So the managers can say, we want 400,000 spawners, and we want to harvest 60% of the fish above the 400,000. So this is harvest rate on the run exceeding the target spawners, okay? And so we calculated a 100-year simulation at each of those sets of conditions. We did 500 Monte Carlo trials for each of those in order to summarize the results that you'd expect in this stochastic world with random variation. So here, for example, for the Yukon River fall chums, we've got average spawner abundance in thousands. And so that's the number that was the average from that 500 Monte Carlo trials, 8,000, okay, top left corner. And the numbers vary across the map. It's much easier to look at a contour map, of course. And what you see is that, as you would expect, as you set the target higher, you get larger average spawner abundance. And of course, when you set the harvest rate lower, you get a larger spawner abundance as well. Now, what's interesting about this example is that you can actually show the managers what the implications are of their settings of their dials by putting a crosshair on here. So this example just says, okay, we have a target of 400,000 and we'll harvest 50% of the fish above 400,000. Unfortunately, there are many other variables that managers think about, and that demonstrates some of the trade-offs. So here we have the same graph we were just looking at, average number of spawners. Up here is the average subsistence catch. Here is the average commercial catch. And here is the percentage of years in which the commercial fisheries actually closed. Now, the green areas show the most desirable state of each of those four indicators. Okay? You want to have few closures as possible, high spawner abundance, high subsistence catch there. So the managers can actually look at this system again with these crosshairs to just say, well, what are the implications of any particular management option? For instance, here, if they want to maximize the subsistence catch, they're going to have a little over 200,000 for the target spawners and a very low harvest rate. But let's say they want to just try out different scenarios. Well, in fact, they would just turn the dials, which you will see before your eyes. And now we have a completely different scenario. Now the manager's thinking, I want to maximize the average commercial catch and put much greater weight on that. Well, what's the trade-off implied? Well, you drop from 180,000 down to 140,000 for subsistence catch. So that's a fair chunk, about 20%. And now you've got more favorable conditions here, lower spawner abundance. So this kind of example of a tool that allows the managers to look at the trade-offs, to quantify them. They know basically qualitatively what the trade-offs are, but not quantitatively. And the issue shown here very well is that these trade-offs are nonlinear. They're not linear. Now, this is actually the simple case, because you look at the broader set of indicators, there are 12 of them. So this top row is for the spawners, so there's median, average, coefficient of variation in the spawner abundance, subsistence catch, same thing, commercial catch, same thing. So you can do the same sort of playing with the management options here. And this brings me to a point that I want to make is I'm working currently with Torsten Moeller and Miriam Boucharian on a way to better visualize this system, because it's very complicated. And their expertise in visual graphics makes for a good opportunity to get past the challenge of trying to convey a lot of complex information. Because not only is there all this, there's another set of layers of these diagrams based on different scenarios or underlying assumptions. And then there's uncertainties in each of those as well. So that is an exciting opportunity to actually look at how we can provide better information for decisions. The next section I want to talk about is where we do some methods from engineering, namely Kalman filter and operating models. So the problem that we're trying to address here is the following. The environment out there is not stationary. That is, there's a changing underlying mean in ocean productivity, in temperature, in all sorts of things. And this means, for example, if there's climatic regimes that are changing, we should take that into account because that's going to affect what actions we take as managers. So here, for instance, is a hypothetical time series of productivity where there was a period of about 20 years here where there's low average productivity, another period of about 20 years of high productivity, and then a longer period of declining productivity. These are quite typical sets of results that we think we've seen in the data. Now, the research objective here was to determine the underlying time trends in productivity. And what I mean by underlying time trends is get away from the high frequency variation that happens from year to year, and the high frequency variation in the estimates that arises from observation error in our spawner abundance estimates, and get down to what are the underlying trends, upwards, downwards, varying, whatever they might be, because those will drastically affect the management responses, or they should anyway. So now, most of what I've talked about up to this stage reflects what most of my research career was up until about 1992, and that is completely focused on mechanistically based research, trying to understand what causes the responses we observe in fish systems. In 1992, I realized, because of that colored diagram cross-section of the ocean that I showed you, there are way too many chains of uncertainty to be able to say what will be the effect of climate on salmon in the future. So you think about it, there's lots of linkages that are nonlinear and lagged between climate forcing and the surface of the ocean currents, and the effects on the biological productivity at the lowest trophic levels, and then nonlinear and lagged and threshold effect relationships between those and fish productivity. So in 1992, I decided we've got to do something else, and I developed the idea that, well, we should have a parallel research program going that explores non-mechanistically based statistical tracking methods to at least try to get as quick a measure as possible of what's happening out there as it's happening. So the challenge in carrying out that desire was that we've got this large observation error, or what I would call noise, on abundance estimates. We've got high frequency natural variation superimposed as well. But we're really interested in the long-term process variation, the underlying signal. Now, it turns out those terms signal and noise should ring a bell, because in engineering, they try to deal with this all the time. And there's a method called the Kalman filter, which is developed exactly for this purpose, to detect the signal amidst noisy electronic data. And it does this by estimating the process variation separately from the observation error. So conceptually, it works like this. In our standard data sets, we try to apply whatever methods we can to estimate productivity, but we have the signal and noise confounded. Whereas the Kalman filter actually separates those two to some extent. So we get a better picture of what the signal is in terms of the changing productivity. Okay, now you might say, well, okay, let's just apply the Kalman filter and go with it. Well, I'm a skeptical sort, so I said, well, no, wait, we have to test it first. We have to ask, is this actually going to work in the salmon system? It might work in electronics. So unfortunately, this kind of testing is rarely done in our field, but we can do it through use of simulation models. So you can imagine a situation where you can specify a true underlying trend, let's say a slowly downward trend in productivity, or increasing, or time varying. It's a bunch of what-if scenarios. And you feed in each what-if scenario into an operating model. An operating model is something that simulates the underlying statistical properties of the system that you're trying to measure. An observation model generates data from the stochastic process or a random process on top of the underlying what-if trend that we specified. And then we feed in those data to the estimation routine. In this case, Kalman filter, and we also looked at other methods as well. And then we compared the estimated values with the true ones that we knew were there to just see how well are we tracking what's going on. Here's an example of some what-if scenarios. So we specified a sine wave with a period of about 70 years, or 60 years, pardon me. Step function, just like we saw in the mid-1970s, there was a huge increase in productivity in the North Pacific that went in a step just like that, and then an autoregressive lag 1 process. These are just examples of what-if scenarios. Okay, here's what the simulation testing showed. The blue curve is the true that we fed in. You notice that the standard estimation method, which is basically fitting the Ricker model as you're used to for those in fisheries, you tend to overestimate the productivity when it's actually low, and you underestimate productivity when it's actually high. But the Kalman filter actually does a very good job of tracking qualitatively, at least, that shape. Same thing with an example here, one run that shows a stochastic AR1 process, overestimating productivity when it's low if you use the standard method, underestimating when it's high. Kalman filter does better. So we repeated that process for lots of what-if scenarios and basically 500 Monte Carlo trials for each of those scenarios to come up with a quantitative measure of how effective each of these techniques is at tracking changes in productivity. The mean squared error of the estimate of A compared to the true one showed a much lower value for the Kalman filter than for the standard method for all three types of scenarios. Okay, so that meant that the Kalman filter passed the test. So it's actually a better method than the standard one at tracking changes in productivity, no matter what the shape of that function is. So we applied that method to some historical data to reconstruct the pattern that occurred to say, okay, how variable has productivity actually been? Once we've gotten rid of all this high-frequency noise, what does it show? Well, here's a stock in Alaska, the Igigik sockeye population, that's gone from fairly low productivity, 2.6 adults produced per spawner, up to a tremendously high value of 17. That means you could harvest 16 out of every 17 of those returns and still have a constant population. Amazing increase in productivity over this 20-year period. The Kwijak population, which used to be the world's largest sockeye population, has gone the opposite direction. Here it used to have relatively high productivity in the early 1970s, and it dropped down to not even maintaining itself in the early 90s, and it's very close to just barely maintaining itself now. So this is something that we wouldn't have been able to see in the raw data, but we've been able to filter out with this Kalman filter method. So there's a clearer signal of the changes in productivity. We've applied the same method to 120 populations that was just published last fall. And we see this tremendous variation that you saw even in the early slides in abundance. Some populations are increasing in productivity, some are decreasing, and they're basically responding to different environmental conditions in different places. So with these better estimates of productivity, we should be able to improve the conservation and management of fish populations, or salmon populations, right? But again, being a skeptic, I said, well, will it? Let's actually find out whether it will by doing some simulations of an entire fishery system. So that brings us to the next phase of this work, which is going to fall under the category of what's called closed-loop simulations. And in engineering, they have a history of trying to develop devices, laptop computers, whatever they might be, and they test them out through quantitative models first, and then they go and build a prototype, and then they test them out, and then they actually find the market for things. Well, why not do the same thing with management regimes and parameter estimation techniques? So the main purpose, though, of this section was to answer that question, will management objectives actually be achieved more frequently if we use a Kalman filter to estimate changes in productivity? And while we were at it, we thought, well, let's go further than that, and let's see if we can identify some management procedures that are robust to a wide range of uncertainties that exist out there that we can't control. And the problem with answering those questions in a very simple way is that there are way too many uncertainties and they're large. So we have to build these models that are closed-loop simulations of an entire fishery, and they're also called management strategy evaluations. That's the jargon used now commonly. Okay, now a closed-loop simulation is nothing different from a flight simulator, except we're doing it in the computer with some fish, computer fish. So if you think what a flight simulator is, it's a way of, it's the size of the corner of this room where they put the pilots in and they lock the door, and then the engineers go in the back room and say, all right, let's throw this stuff at them and see how they respond. So they throw unusual weather conditions, equipment failure, other random events, and through repeated trials, the pilots actually generate a set of procedures that are robust to responding to those uncertainties. So they get to their objective successfully. Those procedures are what we're looking for in management systems for fish as well. So we build closed-loop simulation in this way. We got a little ambitious and we said, okay, now let's not do it for one population. We're going to do it for 15 salmon populations simultaneously. And we're going to have environmental drivers affecting those populations with 3 levels of spatial coherence. None, a large amount, as we see in some parts of Alaska, or a moderate amount, as we see in other regions. We also had various productivity patterns that we specified of the type that I showed you before, the sine waves, step functions, and so on. And there are random processes occurring in all of these cases. Then we generated an observation model, which produced some simulated data, which are of the form time down this axis, spawner abundance, and in adult recruits of different ages in different columns. Those simulated data were fed into 2 different modules. One was a stock assessment module where we tried to estimate the population's parameters. And because we aren't sure which model was appropriate, we used 7 different models to do that successively. Okay? And then we looked at various forecasting models as well, 10 different forecasting models, because forecasts are actually used by some management decision-making modules as well. So here is the management decision, which is composed of a response to the estimates that came out of the stock assessment. These parameters represent productivity measures. And we also asked, well, what if we impose a safety margin? Not just go chasing noise necessarily, but let's say add a 10% safety margin or 20% safety margin on our target spawner abundances. And then finally, all of that fed into a harvesting module. And here we applied 3 different types of outcome uncertainty. Again, none, imprecise, and imprecise and biased. Biased in terms of harvesting too much when the populations are low, which is how typically we see the world unfolding, unfortunately, with the hard data. So all of this is a closed loop simulation here, and it's got lots of complications to it. I can just scratch the surface. What we do is repeat the simulations 100 years across 500 Monte Carlo trials. And we generate results that are of the form that I'll show you now. The first question that I asked was, will the Kalman filter actually improve meeting management objectives? The answer is yes. In terms of long-term harvest, you can see it's higher than applying the standard method. An important finding was that if you turn outcome uncertainty off, you get higher harvest than if outcome uncertainty is on, and in fact, it's substantially higher. So this outcome uncertainty is an important component to include in the evaluations. Now we looked at indicators of conservation concern, that is whether the population dropped below 10% of its unfished value, or average annual catch. So I'll just show you the catch data for the sake of time here. And what you see is for 5 of the different stock assessment models, different catch levels. These 5 models range in complexity from very simple to very complex, going from the standard basic Ricker model to Ricker model with the autoregressive lag 1 term, the Kalman filter, and then 2 of the spatial models, or hierarchical models that I talked about earlier. A non-spatial hierarchical Bayesian model and a distance-based one. The latter is the one that reflects the spatial covariation properties of the populations that are modeled. And what you can see is, even with the most sophisticated model, you only get a 6% increase in the average catch. Now that doesn't sound like much, but it might be millions of dollars, depending on the situation you're talking about. Now that's with no outcome uncertainty. If we then said, what if there's imprecise but unbiased outcome uncertainty? That is, we tend to get, on average, what we set as the target in the management module. There's not much difference between the first case and the second one. In the third case, we impose imprecise and biased outcome uncertainty. That is, we tend to have too high of a harvest rate when the population is low and too low of a harvest rate when the populations are high. These are all empirically estimated functions, by the way. And you'll notice a dramatic decrease in average catch, about 24%. So this is another argument why including outcome uncertainty is a critical element in evaluation of management options. And I should say that the conservation measures did what you would expect them to do. That is, when there's no outcome uncertainty, you tend to have lower probability that the population is going to get knocked to that low level than you will if you have imprecise and biased outcome uncertainty. In fact, in this case, the probability of knocking the populations down to a low level were between 4 and 7 times as high with that outcome uncertainty compared to having no outcome uncertainty. Okay, so from these closed-loop simulations, we can conclude a few things. First, the common filter and other advanced methods that I've shown you help to meet management and conservation objectives by getting a better tracking of the changes in productivity. And I've emphasized here this point that we've got to evaluate management options, taking into account outcome uncertainty. Furthermore, we need to take actions in the field to try to reduce it. The third point, which is more controversial, is that maybe these more sophisticated stock assessment models like that spatial distance-based hierarchical Bayesian model is not worth the effort. It's not substantially better results than the other models. And I'll tell you that was the hardest part to do because those models are so slow at coming up with the estimation. Bridget Dorner, my postdoc at the time, had this model programmed in C++, and we used the WestGrid system to do the simulations. WestGrid is a set of cluster of computers. There are 90 of them here in academic computing services, and I don't know how many at the University of Alberta. And it took usually about a week to run this model on many computers simultaneously. So if it's not getting you very much farther to do with this hierarchical Bayes spatially based model, maybe it's not worth it. And we're yet to get the reviews on that paper, so we'll see what the reviewers say. So the last example I'll give you is applying the concept of diagnostic tests for medicine. So the question here is, how effective are the various criteria that are used for indicating the status of conservation concern for plants and animals, particularly applied to salmon? There are many criteria, many systems developed internationally and nationally. The most common one is the IUCN, International Union for the Conservation of Nature. We'll just talk today about measures of decline, a component of those criteria. There are several others having to do with spatial distribution and absolute abundance. I won't talk about those. So they have variations in how they're structured. The criteria range from having raw data for your abundances to log transform data, smooths with moving averages or not, various other kinds of smoothing. Short term, that is most recent 3 generations, is all you look at. Or do you look at long term by comparing current conditions with some historical baseline, and combinations of all these. So the values that come out from saying how quickly is the population declining over some period are compared to some thresholds that lead to a statement of the status of the stock. So if it's greater than 30%, 50%, or 70% decline, you'll declare it as threatened, critically endangered, or whatever. There are many different categories depending on the system you're using. So the research objective in this project, which was Aaron Porst's, still is Aaron Porst's master's thesis, is which diagnostic criteria for estimating trends in salmon abundance are most reliable for determining conservation status. All of these criteria make biological sense. They're intuitive. But just like you go to a lab, a medical lab, to have a lab test done, they draw your blood and they go do some diagnostic test to see whether you have some disease or not. Those tests have all been developed through large numbers of clinical trials to find out which tests actually have high power to detect correctly that you've got the disease or you don't. Similarly, we should demand a very reliable set of criteria for determining conservation status of populations. So we asked this question of these various methods using a retrospective analysis that is based on historical data. So we looked at 22 different criteria for measuring the decline of salmon abundances. We applied them to 18 different conservation units called CUs. And these were for Fraser River sockeye salmon. And now for some populations we have over 50 years of data. For others we have much shorter data series. And so for each population, we went through and asked in each year, what is the status of the population? So here's an example of applying criterion number 6, which said, has there been a 50% or greater decline in abundance from the first year of data, and using a smooth measure of the first generation of data. The red dots are where that criterion was set off, where it was triggered. It said, yes, there's a conservation concern. So we did this for each of the 22 criteria, so I goes from 1 to 22, each year. And the status of the population was either stated as declining or not declining. We then actually looked at the outcome, what actually happened after that designation by that criterion. Did the population continue to decline or did it not decline? And then we compared the two. And as you've seen in the large variability of those time series, it's not a monotonic trend that we're facing, so it's a complicated question. So what we want is to have a large number of cases of correct designations of the status, either truly declining or not declining. Now, my colleague Andy Cooper in REM pointed out to us that there's actually something that combines these measures into a very simple index of reliability. And it's from the diagnostic method called ROC curves in medicine. So we used that approach and came up with these results. Here's a graph showing for 10 of the 22 criteria the median probability of correctly classifying the decline status of population. So obviously we want that probability as high as possible, right? We want it to be a reliable diagnostic indicator of a conservation concern. We don't want something that sets off false alarms, so we're basically saying, concern, when in fact the population just keep bouncing back. Nor do we want to miss true concerns, right? So this is a probability of those two, of correct decisions in both directions. So the best case was no higher than 0.8. The worst case was down a little below 0.3. The most commonly used criterion is the IUCN criterion A. It's the rate of decline in the last 3 generations, or 10 years, whichever is greater. And it is not the best. It's not the worst. We could do better. The ones that do better are actually those that are based on comparisons of current status with some historical baseline, no matter how far back the historical baseline was. So, in general, all the top rank, there are other various measures we use to say how often did these indicators fall into the top ranked categories. They all point to this conclusion, that long-term measures of extent of decline compared to some historical baseline is by far the most reliable diagnostic tool. The implications of this work are several. I'll just mention one, which is we need to evaluate criteria for classifying conservation status before we use them. We shouldn't just assume, well, this sounds reasonable. I'd like to find out what this indicator tells me. Let's find out through replication how much they actually work, or how well they actually work, pardon me. So the next step in this analysis is to actually conduct some simulations where we can control everything in the dynamics of the population. So if there's any student out there who wants a project, this one's up for grabs. Okay, so I'm going to end now with just 2 quick examples of some other projects that I'm working on with some of my team. And the first one is entitled Guidelines for Designing Salmon Monitoring Programs to Detect Climatic Effects. This is funded by the Gordon and Betty Moore Foundation in San Francisco. It's a conservation organization, and it's based out of UC Santa Barbara, the National Center for Ecological Analysis and Census. And on this team, we have 12 collaborators from various agencies. Kendra Holt, my research assistant, is involved in this project as well. And the product that we're aiming for, we're about a year into the project now, there's another 1.5 years to go, is to produce an online hierarchical knowledge base that will have guidelines for designing monitoring programs for various users. Users will range from highly sophisticated scientists to local streamkeepers groups and everything in between. So it's going to be an evidence-based knowledge base that will provide lots of pros and cons about different designs and resources for designing programs. The second project is based here at SFU. It's the secondary effects of climate change on human and ecological systems, based around a risk assessment approach. This is funded by SFU's Community Trust Endowment Fund. And there are 15 collaborators from various departments on campus, from a wide range of departments. The one example that I want to highlight again, though, is the interaction that we've had so far with computing sciences, because I'm quite excited about this one in particular. It's for improving the way that we visualize complex data. And I learned from Torsten Moeller that one of the challenges in the graphics field in his area is how to represent uncertainties. Not just complex data, but uncertainties in the complex data. And Rob Woodbury is on that team as well. I'm sorry I left off Seat here, Rob. So this is something we're doing as well, and it's ongoing. So I want to leave you with 4 main conclusions or take-home messages. The first one is, I hope I've convinced you that quantitative methods are useful in this kind of work. In fact, I would argue they're absolutely necessary to make progress in our understanding of these complex systems. So we've learned a lot about salmon ecology, and we've developed information that is useful for informing decision makers, whether it's for setting up conservation objectives and actions or regular ongoing management decisions. The second message I want to leave you with is that we need to test the various methods that we use, whether it's simple parameter estimation methods or these diagnostic criteria for conservation status, before we use them. And we also need to test our management options via simulations, these closed-loop simulations. Third point is that uncertainties are always going to be present. We aren't going to get rid of them. So we better learn to live with them and use methods that take them into account as explicitly as possible. And then finally, I hope I've demonstrated that cross-fertilization across disciplines actually works. We're able to make progress that we wouldn't have otherwise been able to, as demonstrated graphically in this slide. So thank you very much for your attention.
Thank you. Questions, please. Steve?
So when you estimate productivity, and then I guess that people could make a decision on harvest based on the current situation, is that estimate, say with a Kalman filter, then is that based on the salmon? I mean, the salmon spiny recruit trends themselves, or is it based on everything like sea surface temperature and every condition you've looked at?
Well, it's a combination of those two, because we have models that are providing a moving estimate of some parameter values as well as sea surface temperature. So some of the models did combine those, yes. Alton?
Randall, for your criteria for conservation status, for predicting conservation status, you had single criteria. You ranked them from the best to the least. Have you ever tried to combine 3 criteria? And it's kind of like using a compass, a GPS, and dead reckoning to kind of find my way in the bush. And each one of those has its frailties, but combined you always get there. So have you looked at that with those criteria?
No, we haven't combined criteria for one reason, and that is many of them are highly correlated with one another. So they aren't really independent measures. Not to say that you couldn't get a better estimate, but we haven't tried that. It's a good idea.
So I like your idea, to test if you can recover the signal from the simulation. For example, you're using try the same function and the step function using to test if a Kalman filter method can work. I know, kind of in statistics, there's a very popular method called non-parametric smoothing. Actually, can recover the signal much better from the what you show in the graph. So have you tried to using that kind of method?
No, we haven't. So that's a very good suggestion. And there are other things that we could have looked at as well along those lines, like neural networks that again compile a lot of information in a non-mechanistic way. And I think that's a good idea to follow up on that, non-parametric smoothing functions. Thanks.
A lot of the methods that you showed were pretty reliant on extensive data sets and things, and I know you've done a bit of work with data poor fisheries, so I'm wondering if you have any recommendations about how you could deal with that.
Well, the only answer I can give is it's situation specific, but in general, I think you need to test out the performance of whatever method you're using with the quality of data that you have available, so you know what kind of confidence to put in the results. So I don't think there's anything generic that I can say in response to that, but it just highlights that need to check out the performance of the methods. Jen?
Is that species specific, or do you think there's you can cross sort of what you learn from one species to a similar species?
Okay, well, that's a good question. So first of all, pink salmon, as you might have noticed from one of my early graphs here, is by far the most variable. and it's got a fixed 2-year life cycle, so it doesn't spread its risk out across different ages like the other species do. So that's one that you're least likely to be able to extrapolate the results from that to any of the other species. The sockeye and chum tend to behave similarly in this respect, so methods that perform well for sockeye also perform well for chum. But beyond that, well, in fact, that raises the other point, get out of salmon. Look at herring, groundfish, whatever you like. Those are quite different systems because they have quite different life histories. And so I wouldn't extrapolate the findings from here, including the findings about how the hierarchical spatial-based model is not very much better than the others. I wouldn't extrapolate that conclusion to groundfish or herring until it's tried. But at least pose the question, that's the challenge. John?
I just had a question about the your testing of the different decline criteria. So under the IUCN system, they're not asking are they declining or not. They're asking if they're likely to go extinct with some sort of probability. They're rather vague, as you know, in the first exactly what that is, unless you use their criterion E, the one which is actually gives a rate of decline, or a probability of extinction, right, after X years. Is that what you evaluated it against, or did you just evaluate it against whether it continues to decline? So in that case, the IUCN criteria are probably even worse. Wouldn't they actually be worse off? Because that's not what they're trying to say. They're trying to say what is the probability that it will become extinct. So clearly if it doesn't continue to decline, the criterion fails. But in fact, it has to do more than continue to decline. It has to decline at some period of time, right, to extinction.
That's right. Yeah, and in fact, the IUCN criterion 2 that I showed you there was, or pardon me, criterion A, which is our number 2, did worse in terms of failing to detect the decline as often as some of those other measures did, but more frequently it was off on the false alarms.
Right, yeah. Okay. And I guess the other problem in actually applying that, of course, one of the reasons though that criterion was developed was because you need minimal information. You don't have to have a historical baseline. Right. So there's always that's another trade-off that we have to deal with.
And in fact, Aaron did redo those analyses using a subset of the 18 conservation areas, 6 of them, which are deemed to be the highest quality data. And indeed, the probability of correctly making decisions went up, as you'd hope it would. And so that reflects that. Some of the data just didn't have as long a time series as others. And there was more uncertainty in the estimates of abundance for some populations as well. Jan again?
Thanks. Just on that topic, those ROC curves, the way I understand them, they're balancing false positives and false negatives. So is there any way to bring in the concepts of the precautionary principle so they're not actually equal? So a false alarm might be weighed differently than a missed species of concern?
Very good point. So in fact, that's exactly what you can do if you take the approach that's used in decision analysis, where you specify some loss function and you say, okay, one type of error is worse than another type of error. So in the case of this conservation index, you want to put a higher cost to missing a true decline and not triggering the alarm than you would for falsely saying there is a concern when there actually isn't. And so we didn't do that. That's a good idea. That's another way to extend these results, or to extend these analyses to get some results that will inform what kind of weighting might be appropriate too. Different people will have different weightings.
So the idea of a closed-loop simulation model sounds very interesting. I wonder if you could comment briefly on how accurate those models are, how many parameters are there in them, and how much data do you have with which to fit those parameters?
Okay, good question. So what I didn't tell you here is that those closed-loop simulations actually are trying to integrate across a large number of hypothesized states of the world. So when you do a closed-loop simulation, you ask, what if the system actually worked like this? And what if the observation errors were like this? And that's why these simulations took so long. You have to loop across a large number of possible scenarios in order to try to map out what the reasonable states are and identify what the most appropriate management responses to those uncertainties would be. So their structural uncertainty is huge. The number of parameters isn't that large, maybe a dozen. The quality of the data that goes into estimating those parameters varies according to the system. You saw some time series are quite long, but you can represent the uncertainty in any of those through a posterior probability distribution easily enough. So I think, as with anything, you always come down to, if you don't use a closed loop simulation to evaluate the robustness of management procedures, what's the next best alternative? And the next best alternative, I think, as we know from experience, is less comprehensive, is less able to recognize these uncertainties that come out of nowhere to upset the expectations. So the El Nino that comes in, or change in climatic regime for decades, as some of the data have shown.
Any other question? Now let us thank our speaker again.
The focus of the SFU Canada Research Chairs Seminar Series is to provide an opportunity to the wider SFU community to learn more about the current research interests of the SFU Canada Research Chair holders. Our next presentation will be on March 26, 2009. Dr. John MacDonald will present his talk entitled "To Be Attended or Not to Be Attended: Recent Research on Attention Capture from the SFU Human Electrophysiology Lab."